It is unreasonable to believe in God in this scientific age. My answer I'm going to give you at the beginning and at the end and probably in between is it's more reasonable than it has ever been in the history of mankind, which is a message you don't hear very often, certainly not in the public media. So I'm happy for the privilege. Thank you for inviting me. And we have a lesson tonight that is focused on my favorite subject, which I know you all share with me, chemistry. Right? The answer is this. And in my mind, the study of that subject is one of the most powerful evidences that has unfolded for us in the last 60 years. I mean, we knew some things before that, but in the last 60 years, amazing increase in our understanding of life at the molecular level. That's my introduction. I do want to read again this evening the theme passage from the Bible, Romans chapter 1 and verse 20. For since the creation of the world, his invisible attributes are clearly seen, being understood by the things that are made, even his eternal power and Godhead, so that they are without excuse. I read that passage because the rest of my time this evening and throughout this series is not going to be spent reading the Bible. It's going to be spent reading God's other book, the book of nature. This passage argues that God has given us sufficient evidence in the natural world, things that are made, to convince us that he exists. I believe that. And I believe we have less excuse than ever for missing that important fact. So that's my introduction, and the argument from design, ladies and gentlemen, is the philosophical argument to which I turn. I focus it with science, but it's the argument from design. Design demands a designer. By the way, this is a class you're allowed to answer when I ask you a question. And I like lots of participation. So let me say that again. Design demands a designer. And magnificent design demands a magnificent designer. I'm convinced. And I think that's what Romans 1:20 teaches. So please take no offense if I now set aside this book and turn to God's other book. So tonight with this lesson, I want to start with you something I mentioned last night. This is the website for the search for extraterrestrial intelligence. And I want to show you what the website says today. This is the up-to-date one. SETI, the search for extraterrestrial intelligence, is an exploratory science. I think I've blown this up. There it is. You can see it better. That seeks evidence of life in the universe by looking for some signature of its technology. So how is SETI going about exploring using science whether there are intelligent creatures out there somewhere else in the universe? How are they going about this? That's what I want to talk to you about. Because, ladies and gentlemen, that is precisely the approach I want to take with you tonight and throughout this series. I think it's perfectly logical to use science to look for evidence of intelligence. But I'm forbidden to do that in a biology classroom. Because they say you're introducing religion into science. That's a system of faith. Science is a system of facts and reasoning. And yours is religion. Well, are these guys religion? I don't think so. Most of these folks are not believers in God. But they're using science to look for what? Were you with me? What are they looking for? Evidence of life. By the way, intelligent life. Because what they're hoping is somebody's going to say something to us. By looking for some signature of its technology. So how do you go about looking for signs of intelligence out there in the universe? And then I'm going to say to you, how do you go about looking for signs of intelligence right here under your nose? Whatever they're doing out there, let's use science to do the same thing right here. Virtually all radio SETI experiments have looked for what are called narrow band signals. These are radio emissions that are at one spot on the radio dial. Did you listen to your radio coming over tonight? You dialed a 95.6 and you hear top 40 or whatever. But you can get over the radio dial information, can you not? Signals that speak to you or that sing to you. Intelligent information. Well, that's what they're looking for. Narrowband signals, they say, are the mark of a purposely built transmitter. Are you with me? Purposely built. May we say it another way? Intelligently designed. Their position is, if you find narrowband signals that contain information in them, you have found a signature of intelligent life. I don't want to argue with that. If they were to find such a thing, that would be powerful evidence that somebody out there is intelligent. Because nature doesn't do that. Separate from intelligence. I agree with their argumentation. The problem is they haven't found one iota of evidence that supports what they're looking for. So what is the signature? They're looking for narrow-band radio signals that contain information. And were they to find that, they would consider that a signature that there's life out there, intelligent life in the universe. I hope you got that, because that's only the foundation for everything I'm going to say tonight. Here's another very important point. SETI can infer intelligent cause without identifying what that intelligence is. Are you with me? Think carefully about that. Just because they found some radio signal with information, it doesn't mean they know much about that intelligence out there. But they are convinced by that there had to be intelligence behind this. Folks, there's a limit to what science can tell us about God, because He's beyond observation. Only by indirect observation. Well, that's exactly what they're doing. They're looking for indirect observation. There's something intelligent out there. So you can infer intelligent cause without identifying what that cause is necessarily. What if we received a message from space with a language in it that we could decode and read? How about one that said, "Help! Come find me!" And you could read it. If you really found such a signal, I doubt there's anybody on earth that would say, "Well, isn't it interesting how the stars bleeped that out?" I don't think so. What if we got a message from space with a language in it that we could learn how to read, decode it? What we have, ladies and gentlemen, at the microscopic level right here on earth, not out there somewhere, but right here, is evidence of a signature in every cell of every living thing. There are signs of intelligence right here on earth that people are overlooking in the scientific community. Every living cell is full of signs of intelligent design, I would argue, including a language which we've learned to decode and read. Every living cell has a language in it. Did you know that? Here's an interesting gentleman you may have not been introduced to. His name is Antony Flew. He is, and was from the time he was 15 years old, an atheist. In fact, he was an avowed atheist who was willing to argue his case against creation and God for 50 years. He debated lots of creationists. including Thomas Warren. Do some of you know that name? Thomas Warren worked at Harding College and was a philosopher. And he debated this man many years ago on the subject of God versus not God. And this gentleman, Anthony Flew, took the position there is no God. Now, do you see the book over here on the left by Anthony Flew that was written after he turned 80 years old? And you'll notice how he marked out no and replaced it with a, because Anthony Flew has become a believer that there at least is a God. He doesn't believe the Bible like you and I do, but he's at least become convinced there has to be some kind of supreme intelligence behind this. Well, what was it that finally turned the page for him? It now seems to me, he said, that the findings of more than 50 years of DNA research have provided materials for a new and enormously powerful argument to design. So said Anthony Flew. Folks, I appreciate a man that at 80 years old will change his mind about something as significant as that. I hope I'm open enough to learn new things. I'm 74, almost 75. I want to keep learning, folks. And I hope I'm open enough if I learn something that's different from what I believed all my life, if it's true, that I believe it. I hope you are too. This man, in the vociferous criticism of his friends who are atheists, took a stand. Amazing. You need to read the book. There is a God. Worth reading. Here's what he said. I had to go where the evidence leads. So my job tonight in 2 lectures is to overwhelm you with the evidence of the cell at the microscopic level. And I'm thrilled to get to do that. And I hope you're thrilled to join me. And if you're not, stick with me anyway. So I'm going to lean on this book a good deal called Signature in the Cell. That was no accident that he called this book Signature in the Cell. Because what did SETI say? They're looking for some signature of intelligent life in the universe. And he's saying, it's right here under your nose, and may I add, in your nose. Because every cell in your body is shouting intelligent design. That's what this book is about. It's a little thick, right? Stephen Meyer is an amazing writer in my view. He's a scientist. He's also a philosopher, but he's a good writer too. You'll enjoy this book, and if it gets too heavy chemically, you can skip that part and still get the message. It's worth reading. So I'm going to lean on that book and some others as we go along, as you will see. And the question will be, which is more reasonable, ladies and gentlemen of the jury? You will be your own decider about this. You're going to look at the evidence of nature. I'm going to give you evidence, things that we have learned about our natural world. And then you're going to have to decide which is more reasonable, natural causes, no God needed, or as I told you last night, Dawkins says, all you need is the blind forces of physics acted upon by natural selection. And you can get all the appearance of design in the whole world, no matter what it is. It happened that way. No God, foolish to think there is a God, dangerous to believe in God, that's one position, or intelligent design. Which is more reasonable based on what I'm going to show you. So here we go. What is life like? I'm talking about the fundamentals of life. So I thought I'd share with you a piece of a book by Francis Crick. Now, who in this audience knows what Francis Crick is famous for? What's he famous for? Exactly right. Francis Crick and James Watson are credited with discovering the structure of DNA. Anybody know what year that was that this was revealed? 53. How'd you know that? Good for you. Have you read the whole book? Not yet. It's a great book. 1953. How many in here were alive in 1953? Look at this. Thank you. I was too. I was 8 years old. What a dynamite year that was. They uncovered the secret to DNA. I'm going to tell you about that a little bit. He later wrote a book called Life Itself, Its Origin and Nature. The origin and nature of life. So I'm going to share you a little bit from that book. And up here on the screen, you'll see a little summary. What is life like? Life as we know it on earth, and can I pause there just a minute? Life as we know it on earth, folks, is the only life we know anything about. They like to say life as we know it as if there's some other life forms out there. We have zero evidence of any other life forms. Got it? Life as we know it on earth appears as a synthesis of two macromolecular systems. May I put that in common English? Two systems of large molecules. And we'll break it down here. Life has structural materials, that is, proteins. They make up for the shape and mobility of things. Every living thing has that. Second, it has tools and machinery. They're called enzymes. But folks, enzymes are just special forms of proteins. So both of these types of materials are proteins. And we're going to talk about that soon. They make up the metabolism or the growth and maintenance of a living thing. If that's all you had, you'd have no living things, folks. It requires another major set of molecules that are called the blueprints. They are the genes, the nucleic acids, the DNA and the RNA of living things. And they allow for reproduction. They contain the information and the directive function to allow you to reproduce yourself. Every form of life that we call living has those two great classes of molecules, proteins and and genes or nucleic acids. And the proteins, as you can see right here, are assembled from amino acids, which we'll talk about in a minute. And by the way, there's a quiz in the morning. Because of their versatility, the proteins do all the work, but are unable to reproduce themselves in any simple way. They do the work, but they can't do this. The RNA and the DNA, let me pause and go to my next slide. Two proteins, structural and enzymes, are the workhorses. Isn't that what I just said? And this is what they're made of. The 20 common amino acids. So class, shall we go through this? You're in my chemistry class. If we had started on our biochemistry section, this is the first thing we would give attention to with regard to proteins. Because every protein in every living thing is made up of this. So let's go through it here. Glycine, abbreviated GLY, alanine, ALA, valine, VAL, and so on. Leucine, isoleucine, serine, glutamine, arginine, tryptophan, histidine. These are the 20 common amino acids. Every protein, ladies and gentlemen, every living thing, from single-celled organisms to us, are made of these 20. Now, I've been corrected by medical students to say, well, there's some few exceptions to that. So, okay, 0.0000001% of some of that stuff has an extra thing in it or two. This is good enough for government work. Pardon me. The fact is, the common amino acids, these 20, make up the proteins that are in your body and in every other living thing. It's very much like the 26 letters of our English alphabet, which make up all our words. Right? You agree? The word throughout, Throughout, that's a combination of the 26 letters of the alphabet, right? How about anti-disestablishmentarianism? Still a combination of the 26 letters of the alphabet. Every protein, get this clearly, class, is made up of a combination of these 20 stacked in a row. Okay? That's a fact. Nobody argues with that. We've done it. We've identified proteins and told exactly what the sequence of amino acids is in every one of them. So that's the proteins, that one great class. Now we're back to the DNA and the RNA, and I want to read you what Mr. Francis Crick said. I didn't say this. Are you clear that that's not what I'm saying? He says RNA and DNA are the dumb blondes of the biomolecular world, fit mainly for reproduction, but of little use for much really demanding work. I'm sorry, ladies in this audience. I didn't say that. But I say it here because it's probably the one thing you're going to remember. RNA and DNA are fit for reproduction, but not much else. So listen closely. The problem of the origin of life, and what he means by that is, how did life get started here on earth without God? Nature just did it. How does that happen? Here's a huge problem. It would be a great deal easier to approach if there were only one family of molecules capable of doing both jobs: reproduction and catalysis, all the work. But life as we know it always employs two. You have to have them both. And by the way, you have to have them both functioning together at the same time. We know of no life form that doesn't have both of them functioning at the same time. How does that happen? He goes on in the book to talk about the challenge of this. And a little bit later in the book, he talks about how we might need to look somewhere else than Earth to have enough time to produce living things. And he's counting on 4.5 billion years on Earth. But he knows well enough about life that that's probably not enough time, given all the complexities we're going to talk about. So then he spends the last several chapters of this book talking about how somebody sent this here on a spaceship. He calls it directed panspermia. And he talks about what the spaceship might have been like and what they might have sent on that spaceship to seed the first forms of life, which probably wasn't enough time here to produce. This is a Nobel laureate, ladies and gentlemen, in chemistry. And what evidence do we have of a spaceship sending life forms here? Absolutely zero. But I'll spend half of a book on how life got started talking about that rather than saying there's lots of evidence for intelligent design. Anything but God, folks. Nobel Prize, you read it for yourself. Half of this book is about that mess, for which there's zero evidence. Amazing to me. The facts are DNA and RNA are reproducers. Proteins do lots of work. You can't get life without them both. It's a huge problem for how life started without a designer. Sure looks like a design to me. The structure of DNA that Watson and Crick uncovered in the 1950s is a characteristic structure that they called a double helix. Look over here. You can see this strand wrapped around that strand. Can you see that? And then there are cross pieces that connect the two strands together. And because of that, it has the capacity to reproduce itself, to copy information and use it for other things. Now, all of you students here on the front have studied this, right? At least to some degree. And I'm telling you, audience, this has been known since the 1950s, so this is no new information. And people have accused me of being really dated, which is part of the reason I'm doing another lecture after this one. Because most of what I'm going to talk about in this lecture we've known since the 1970s and early 80s. But that's okay, you need the background. So DNA has this unique structure, and there's a long history that Stephen Meyer gives in here that's fascinating about how they finally uncovered this. Have you read that part? It's fascinating. Because he's also a historian. He tells you some of the background. So here's a DNA molecule, a picture of one. You see over here it's wrapped around itself and see these cross pieces. And the DNA actually unzips. And it exposes this side and that side. And look along this side. You see there's an A, C, G, T, A, G, G, T, A, C, G, so on. And by the way, when you expose that DNA, there are no letters sitting there. There are chemicals that sit there and are exposed. We call them, can you kids help me? What does the A stand for in DNA? Adenine, C, cytosine, G, guanine, and T, thymine. There are 4. When you look down this entire list, you're only going to see 4 letters. Because there are exactly 4 bases inside the DNA that have the code in them. This language has 4 letters. And in this language, ladies and gentlemen, the 4 letters are used 3 at a time. Every word is 3 letters. Now, wouldn't that be an easy language to learn? Only words you have are and thee, and sat, and cat, and bat, 3-letter words. But ladies and gentlemen, the fact that tied up in this molecule is a language that we have learned to decode and read. We know exactly what it means now. It's utterly amazing. What do you know in nature that will produce a language by itself without intelligent design? I simply ask you. I would say, if this were a jury, ladies and gentlemen, my case is closed. We have a language in every living thing. But that is not the end of the story. It's just barely touching the beginning. So, you can see here that what happens is, as this is unzipped, and by the way, you see this right here, RNA polymerase? If a word in biology ends in ASE, that means it's an enzyme. You saw that before on the previous screen because you're really with me. Do you remember that an enzyme is just a special case of a protein? Good. Now I'm going to tell you something, class. I'm going to be asking you several questions over the next while. The answer to almost all of them is proteins. So you know the answer, right? Before I say it. So, this molecule right here that you see, RNA polymerase, is an enzyme, which is a? Exactly right. The protein rides along the DNA and allows it to unzip and read it and then make copies of it. RNA is a copy, a transcript of the information on the DNA. So, all right along here, you're forming the copy. And that copy then can be used. So the information is taken somewhere else and used to do useful things. Amazing. And there, ladies and gentlemen, is the language. You can put this language on one page. It's called genetic code. And it's called that because it's a combination of 1, 2, 3 amino acids in a row. And not 1, 2, 3 bases in a row that make the amino acids. You recognize any of those names up there? You were supposed to memorize the chart I had, the 20 amino acids. Because tomorrow, if you were in my class, we'd have a quiz on the 20 amino acids, and you'd name them and give me their abbreviations. And the next day, you would give me their chemical structures. Very important, because every one of them has a unique structure. Why are they called amino acids? Because everyone has an amine in H3 group, and everyone has a COOH group, which is an organic acid. So it's an amino acid. But they're all different. So you got that now, and you've memorized the 20, so we're ready to move on. What you see up here, class, is all 20 of the amino acids that have a code. So let me teach you how to read. If you read U, that's 3 letters, right? U. You know how to pronounce that word? Ooh. This is not a spoken language, folks. But U is a word. And you say, why is it U? Well, I don't have time to explain that, but in RNA, the T is replaced by a U. It's called uracil. And that's another whole subject we'll talk about some other time. So what I have up here is the RNA language. So if you read the word U, what does that mean? Here's what it means. Phenylalanine. Let's try another one. You tell me. What does C mean? Proline. Got it? So you can read every 3-letter word up here. I might ask you a math question. If you have 4 letters to use and you can repeat the letters, and you can only use them 3 at a time, what's the maximum number of words you can make? 4 letters taken 3 at a time with repeats allowed. The answer, ladies and gentlemen, is 4 times 4 times 4. Do the math in your head. 64 words. There are exactly 64 3-letter words in the genetic code, and they're all up here. Do you see some repeats? Yeah, because there are only 20 amino acids, right? So you have more than one code for proline. There's 4 of them right there. 1, 2, 3, 4, is that right? Yeah, 4. Here's 4 for threonine. Is asparagine. There's isoleucine couple there, and leucine's got a bunch. Looks to me like a backup system, ladies and gentlemen, in the code. But every amino acid's coded up here. In addition to that, you got to know where to start reading. So there's a code for that. And you got to know where to stop reading. Do you see up here there's 3 codes for stop? Those are called terminators. And when you're reading along that strand, you saw when it was unzipped, there was a strand to read. When you get to one of those 3 codes, you stop reading. So watch me closely, everybody. As you come along that strand, whether it's RNA or DNA, you got to start reading somewhere. That's the code AUG. And then you read 3-letter word, 3-letter word, 3-letter word, 3-letter word, and you get to one of the stop codes and you stop reading. And in between that, you have a string of 3-letter words that make up amino acids. And amino acids in a string make a protein. Exactly right. So what you have, ladies and gentlemen, inside every DNA, inside every cell of every living thing, is a code for every protein that's got to be built. I pause. Because a lot of teachers teach that as if there isn't anything amazing about that. You ought to be going. That's not what happens in our age. We kind of brag on how we've discovered all this stuff. The question is how to get there in the first place. I ask you, class, what if we received a message from space that had the genetic code in it? And we could figure it out from the message we got from outer space. What do you think everybody would say? Of course they would. SETI and any other reasonable people would say this came from an intelligent source. The stars didn't bleep out the genetic code. But when it's in every living cell, they say, isn't that interesting what nature did by the blind forces of physics? That's exactly where we are. We have much more evidence right here than we do out there for intelligent design. Ladies and gentlemen, this is amazing. But this hasn't touched the hem of the garment of what's happening inside every cell, because not only do you have a language, you have that language doing something at a remarkable rate every second. And here's a little summary of part of what's going on. You see, here's the DNA with all the information in it. You see it unzipped here and a copy being made. Are you with me? And that information then, having been copied, you take it down here, and this is called the messenger RNA, and it is edited. You see these little white pieces? It's edited. The introns are snipped out, and it's reorganized, and this is called the mature messenger RNA. And then it migrates. You have to transcribe, that's this piece. You have to migrate, and then coming outside the cell, or the nucleus of the cell, it comes over here to these two ducts hugging each other. Those are not ducks. They make them look like ducks. This is called the ribosome. And the information that was taken from here and migrated is read in the ribosome and ends up producing a protein. And may I say something to you right here? Every step in that process requires proteins. And what are you making? Proteins. In fact, the process I've just described to you here takes at least 50 different proteins to make it happen. And all 50 of those proteins are coded in the DNA and have to be made themselves. I lost about half of you on that one. The DNA has all the information for every protein, but it takes proteins to get the information out and make proteins. So how'd you make the first protein? Without having the system already in place that was designed. How do you have it? It's a big old problem, ladies and gentlemen of the jury. So now, can you see that all right? Hold up just a second, techie guy. Stop. Brian, stop. Can you see that well enough, or do we need to dim the lights a little bit? Let's dim them just a little bit. Not enough for you to go to sleep. Brian, can you look at there. Can you call on this man and he just... All right, let me get out here now. This is in the website for the book, Signature in the Cell. You can go watch it. And there is an audio behind it, but I'm better. So I'm going to be the audio. He has his own audio. I'm going to do mine. So here we go. What you see in front of you, ladies and gentlemen, is a mock-up of the DNA molecule. And you can see here the different letters A, C, G, and T inside of it. So let's keep going now, Brian. Uh-oh. We're trying to stop and start this video clip. All right, so here we go. Let's see if we can make it. Here we go. All right, so as you watch that beautiful DNA strand, and you ought to understand this is a cartoon. Let's stop right there. You see that glob? I told you in the last section that's RNA polymerase. And you remember anything that ends in ase is an enzyme, which is a protein. Can you see the different colors up here? There's several different proteins actually that ride along that DNA strand. Several proteins, not just one. And can I take a little side trip here? One of those proteins is for the purpose of not allowing the DNA that unzips to get so messed up it can't zip back up. Have you ever had problems with your zipper? DNA is so complex when you unzip it and it's all twisted around. If you don't have something that it can keep it able to zip back again, you got problems. So there's a special protein to help it zip back up. So there's a glob. These are proteins doing lots of things, but they're riding along the DNA and reaching a point now where they're going to unzip it and allow it to be read. So let's go, Brian, here we go. All right, you see it flapping its ears there? All right, stop right there. This is what it looks like. It's more like that. This blob looks more like what this is. And down inside, the work's taking place as it unzips the DNA. And you see this little thing right? This is the RNA coming out the side. Okay, now let's go down inside this blob of protein that's wrapped up around the DNA. Let's go. All right, there you are. We're down inside. You see how dark it is down in there? And we are unzipped. Stop right there for just a second. And what you're seeing here is you see these exposed chemicals here. They're A, C, G, and T, you remember? In a certain order. And what's happening is we're bringing up one piece at a time to match what's on the DNA. Kids, tell me, A always matches up with T, and C always matches up with G. So it's like love and marriage and a horse and carriage. Always A and T match up and G and C match up. So if there is an A on this one, there's going to be a T on this one. And if there's a C on this one, there's going to be a G on this one. You always get the exact match. And if you were in my chemistry class, ladies and gentlemen, you would be able to explain to me why it is that A and T can match up and C and G can match up and not the other way around. Because it's chemically designed. By Dawkins, by blind forces of physics. Believe it if you want to. So the copy's being made now. Let's go ahead, Brian. One of the problems with this cartoon, as it makes this copy, see the RNA being made as you go along there? Stop again, please. That was way too slow, folks. Way too slow. A typical protein in living things like you is 1,000 amino acids long. 1,000. Which means you've got to have copied 1,000 of those amino acids before you stop. Typical. You have lots of proteins in your body, thousands of proteins, different ones that are all being made. One of them typically is 1,000 long. You know how long it takes to copy those 1,000 amino acids in the code? Watch me. That was about 10,000 times too slow. Your body is copying amino acid codes at a rate of millions per second every minute you live. That's why you better eat your proteins, and that's why you get tired sitting here watching me. I'm doing all the work. No. All right, so we've now made the copy of the one DNA copy for the one particular protein, and the RNA is swimming now as it migrates. Go ahead, Brian. And by the way, you remember it took a bunch of proteins to make that happen. So here it goes. Now it's coming to the cell wall. Stop right there. It's coming to the cell wall of the nucleus. See these holes? There's lots of holes in the cell wall of the nucleus, and it allows things in and out. Do you think any respectable nuclear cell wall allows just anything in and out? No, ladies and gentlemen. Only certain things go in and out. So here comes this RNA, and it comes up to the door, and watch what happens. Here you go. You see this thing right here? That is, ladies and gentlemen, a what? Protein that opens the door and says, come on through. It's a guardian protein. Stop right there. You see here on the other side, whoops, we missed it. Back over here, there are holes. There's another protein on the other side. There's a protein on both sides. So you don't just get in and out without permission. So there's a very specialized protein that allows the RNA to migrate outside the nucleus and not other stuff. Okay? So add that to your list of. Got it. All right, now it's migrating. Let's see what happens. Go ahead. Here it comes out into the cytoplasm of the cell, and it is picked up by these two ducks hugging each other that looks more like that. Those are ribosomes. And they close in on it, and down inside, stop right there, please. Down inside those two ribosomes that took that RNA inside, here's the code from the RNA that came right off the DNA. Now you're reading that code, and see that CAC right there? That is a specific amino acid. And the amino acids are floating around in your cytoplasm of your cells. There's a specific RNA called transfer RNA for every amino acid, of which there are 20. So there's 20 transfer RNA acids, and there's a protein for every one of them to make all this happen. So that's 20 more proteins. So the transfer RNA, see these guys here, doesn't that look like a, what does that look like? Kind of like a seahorse, doesn't it? And down at the bottom, it's got the amino acid on it, and up at the top, it has the specific code that is the exact mirror image of that code. So this code is what's in the DNA, and it matches that one. And so once this one is there, this one comes and joins it, and then these two amino acids hook up. Now you have two amino acids in a row. Then the next one comes up and hooks up to those three letters and joins this amino acid to those two. And then you have three amino acids. Right? A string of amino acids makes a protein. So a protein is being built here, ladies and gentlemen, by this method. All right, now you keep going, Brian. A problem with this one is also it's way too slow. Way too slow. Come on, move it. Shape up. Faster. Faster. This is way too slow. It's pitiful. Do you see what's happening down here? It's building a protein, and they're trying to make it fast enough. Stop. Here's the messenger RNA, and you can't see it very well, but you see right there that little blob coming out the bottom? That's the protein coming out the bottom of the ribosome that came from the code, that came from the DNA in the nucleus that was built by this method. May I stop right here, class, and ask you, can you in your wildest imagination dream up that this happened without a designer? For all kinds of reasons. But we're not finished. Because look, a string of amino acids makes a protein, all right, but it is not finished. Because the protein can't just be a string of amino acids. It's got to be shaped in a specific three-dimensional configuration. There's primary, secondary, and tertiary order to every protein. And until you've tertiated them, if that's a word, you don't have a functional protein. So do you see it's kind of globbing up down there? Keep going, Brian. Protein's right here. Kind of globbing up. It's going to release it eventually because it's got exactly the right number of amino acids, and then it migrates to another organelle in the cell, and it goes down inside so that it can be properly shaped, and you can watch it here as it shapes into a specific shape for that protein. And the reason it can do that, ladies and gentlemen, stop right there. It does not turn white when it gets finished. That's an effect. But the reason it can do all those things is because the types of bonds, chemical bonds that are in that molecule that allow it to do all that reconfiguration, and it comes out precisely in the shape needed for that particular protein to do its particular job. And then it is released into the cytoplasm, and guess what the authors say when they describe this? To do the job for which it was designed. And then they say, but it wasn't designed. Go ahead and finish it. So there it goes out into the cytoplasm. and it will go to the specific site where it needs to be to do the work that it is designed to do. Okay, Brian, thank you. That was very good. Thank you for your good help, and if you'll take me back to the slides now, that would be great. Transcription, ladies and gentlemen. The cell copies the information. It requires at least 20 different to make that happen. Then the messenger RNA is snipped and matured. Guess what it takes for that? Yeah. You have editosomes and spliceosomes that are proteins to make that happen. And when the migration takes place out of the nuclear cell wall, guess what it takes? Proteins. And then you get outside the cell wall and there's a bunch of more proteins required. Every transfer RNA, and I haven't even talked about these guys, brings in specific amino acids, and every one of them has a protein specifically for that to make it come in here and make that copy. A living cell is a marvel of detail and complex architecture, says guess who? Whoops, let me go back. See that? Carl Sagan. 1974 in Encyclopedia Britannica, which is a big old book with hardly any pictures. Seen through a microscope, there's an appearance of almost frenetic activity. You know what frenetic is? Watch. On a deeper level, it is known that molecules are being synthesized, that means built, at an enormous rate. Almost any enzyme, which is catalyzes the synthesis of more than 100 other molecules per second. So since you've been sitting there, every second, every enzyme in your body's been helping your body produce 100 other molecules. I'm tired. In 10 minutes, the sizable fraction of the total mass of a metabolizing bacterial cell has been synthesized. That means it's been rebuilt. No wonder bacteria is dangerous. Every 10 minutes they reproduce themselves. The information content of a simple cell, forgive him for that language, has been estimated as around 10 to the 12th bits, comparable to about 100 million pages of Encyclopedia Britannica. This is an information machine. And isn't it interesting how the blind forces of physics built a Britannica encyclopedia of information inside the DNA and the functioning cell? You can believe it, ladies and gentlemen of the jury, but I would suggest you have greater faith than anybody I know. Over 200,000 molecules are being built per second in every cell you have. If we received a message from outer space containing just the genetic code and an explanation of how it is used to build proteins, everyone would conclude the source was intelligent, would they not? Listen, folks, if we got a message from space with the genetic code and a brief explanation of how it works to build proteins, it would be in every headline of every newspaper on earth. We have had somebody out there tell us intelligent stuff. Not. The blind forces of physics, aren't they amazing? Everywhere we look, says Michael Behe, from the macroscopic to the microscopic, things look like they're made. There's a loud, clear, piercing cry of design. Get this book. I'll be using it tomorrow. And that's the end of my lesson at this hour. And we're going to have a what? 7-minute break. I stopped in an hour, folks. Let's be back in about 7 minutes and we'll start lesson 2. And let me tell you a little preview. I'm going to throw a monkey wrench in all of this.